This is the second part of lecture 7, and uh, in this lecture we learn about the three domain classification system. So the three domain classification system is based on the similarities of ribosomal RNA, if I say more specifically 16S ribosomal RNA and organization of DNA. So based on the similarities of 16S ribosomal RNA and organization of DNA, all the microorganisms, they fall into three domain, okay, according to this classification system: domain Eubacteria, domain Archaea, domain Eukarya. So first, the domain Eubacteria, they are prokaryotes with looped ribosomal RNA and peptidoglycan in the cell wall. All the known bacteria, Streptomyces, Actinomyces, Cyanobacteria, are in this domain. So domain Archaea, they are prokaryotes with non-looped ribosomal RNA and are characterized by extreme tolerance of methane, salt, temperature, and acidity. Methanogens, acidity. Methanogens, halophiles, picrophiles, and pyrophiles, they fall in this domain. So sometimes actually, as these microbes, they can tolerate extreme environmental conditions, so that's why they are also called extremophiles. That Archaea, they are also called extremophiles. Then finally, domain Eukarya. Domain Eukarya are eukaryotes with sorry. So domain Eukarya are eukaryotes with non-looped ribosomal RNA. They have 18S ribosomes and membranous organelles. All the fungi, protozoa, algae, plants, and animals are in this domain. So this slide, it shows the this three domain concept of the three domain classification concept, and also tell more about the microbes that fall in this each domain. Okay, so like if you carefully look at this classification tree, okay, so think is if you look at this figure, you will see some arrows. Okay, like for example Archaea. Okay, so the first, the life form, they begin to evolve in this earth around 3.5 billion years ago. Okay, and and then if you think about the Archaea, so and if you look at this arrow, then you see that first they told the hyperthermophiles, then methanogens, extreme halophiles. So this arrow, okay, these are actually also represent the time when these microbes get evolved. That means, so according to that, if we look at the Archaea domain, hyperthermophiles, they were evolved fast. Okay, then the extreme halophiles and methanogens, they evolved almost in the similar time. Okay, so similarly, the bacteria, in case of domain bacteria, uh, Thermotoga, gram-positive bacteria, cyanobacteria, gram-negative bacteria, chloroplast, and mitochondria. Well, we can also see mitochondria and chloroplast here, but are mitochondria and chloroplast individual microbes? No. So why they are in this domain? Okay, I'll talk about it later. So then, first, let's see the eukaryote. Okay, so eukaryote, you can see, the evolutionary tree here. So first, Archaeozoa, finally fungi, animal, plants, etc. Okay, so why mitochondria and chloroplast here? Okay, and what this arrow, this red arrow and green arrow means? Okay, so basically this three domain concept, it also accepts the endosymbiotic hypothesis. The endosymbiotic hypothesis, which was first proposed by five kingdom classification system. That means which was proposed by Whittaker. Okay, so and what we know, if you remember, the endosymbiotic hypothesis means that eukaryotic cells, they evolved from prokaryotic cells. So it was believed that once the mitochondria and chloroplast of the eukaryotic cells, they were actually prokaryotic cells. Through endosymbiosis, they become part of the eukaryotes. That means different, like for example mitochondria and chloroplast, they are two different prokaryotes. So they begin to live in this endosymbiotic relationship, and eventually through the evolution they become eukaryotes. Okay, I'll talk about this thing, endosymbiotics, in more details in later slide. Okay, so anyway, so this next slide, it shows the comparison between this three domain based on cell type, cell wall, membrane, and lipids. First amino acid is in protein synthesis. For example, if we think about the cell type, archaea prokaryote, bacteria prokaryote, eukaryote, eukaryotes. If we think about the cell wall, the cell wall composition of archaea varies. Okay, however, they don't contain any peptidoglycan. But cell wall of bacteria, they contain peptidoglycan. Again, cell wall of composition of the cell wall of eukarya, they varies and contains different carbohydrates. And if we then think about the membrane lipids, archaea composed of branched carbon chains attached to glycerol by ether linkage. But in case of bacteria, the membrane lipids composed of straight carbon chains attached to glycerol by ester linkage. Okay, ether linkage, ester linkage. And in case of eukarya, the membrane lipids are composed of straight carbon chains attached to glycerol by ester linkage. Okay, and the first amino acid in protein synthesis, in case of archaea, methionine, in case of bacteria for formyl methionine, in case of eukarya, methionine. Okay, so now, I mean, in the last slide I talked about the mitochondria, chloroplast, and the endosymbiotic. So what is the explanation? What is the explanation of existence of chloroplast and mitochondria in eukaryotes? So you can see that chloroplast and mitochondria were here, but by this arrow they were also shown in eukaryote domain. So what is the explanation? Okay, so some scientists think the organelles of eukaryotic cells evolve from endosymbiotic prokaryote cells. Like the mitochondria and chloroplast, they are organelles of the eukaryotic cells, right? So, and some scientists thinks that these organelles of the eukaryotic cells, once they were prokaryotes themselves, and through evolution, which is called endosymbiosis, they becomes organelles of the eukaryotic cells. Okay, and this evolution occurs during the 1.4 billion years of endosymbiosis. Okay, so and during this endosymbiosis, these prokaryotes, they lost their individuality and adapted to exist as organelles only. So some scientists think that, but what is the basis? I mean, this is science, right? You cannot see, you cannot tell anything without logic. So what are the logic behind this? That scientists are claiming that they're saying that I mean, the organelles such as the chloroplast and mitochondria, they became part of the eukaryotic cells through endosymbiotic, that is through evolution. Okay, so the logics behind this are both chloroplast and mitochondria have 70S ribosome, circular DNA, and are sensitive to chloramphenicol. So these features, they are features of prokaryotic cells. Prokaryotic cells also have 70S ribosome, circular DNA. Okay, so that means these features of chloroplast and mitochondria are similar with the features of prokaryote cells. So which means I mean, it is highly likely that chloroplast and mitochondria, once they were individual prokaryotes, but through endosymbiosis they become organelle of the eukaryotic cells. Okay. In addition, chloroplast and mitochondria divide independently of the eukaryotic genomic DNA. Okay, I mean, not only this, like the both chloroplast and mitochondria, they have 70S ribosome, circular DNA, sensitive to chloramphenicol. These are not the only logic. Okay, so additional logic is both chloroplast and mitochondria, they divide independently of the eukaryotic genomic DNA. So by this logic or by this evidence, the scientists presume that they presume that the eukaryotic cell or the organelles of the eukaryotic cells, they were evolved from pre-existing prokaryotic cells. Okay, well, now finally we know that microbes are very small, right? They're very small. So does this smallness provide them any benefit? I mean, does this smallness have any significance? Yeah, of course this have. Of course there are. Okay, so I mean, as the microbes are very small, they get some benefits because of this smallness. So let's try to get more idea about this. So what are the significance of smallness? So what kind of benefit the microbes get because of being so small? Okay. So first, the very small size of microbes has an impact on fitness as it affects metabolism, generation time, colonization ability, and growth rate. Okay, so as microbes are very small, okay, their metabolism is faster. They can generate, that means multiply in a very short period of time. Okay, they can colonize easily. Okay, and because of this, their fitness is affected. Basically their fitness is affected towards a good thing. Okay, I mean they are more fit. Okay, and then a small size means relative to cell volume more surface is exposed to the environment because they are very small. So more surface is exposed to the environment. As more surface are exposed to the environment, nutrients and waste products, they can rapidly pass into and out of the cell as transport distances of a metabolite within an organism and between the organism and the surroundings are short. Microbes are very small, right? So for example, think about the very big very big organism. Okay, think about the very big ball. Okay, think about the football is big, and now think about the table tennis ball is small, football is very big. So consider this as organism. Okay, and so football is organism, table tennis ball is an organism. So in case of football, so if I mean say in the middle of the football there are some metabolites and which need to transport it outside of the cell. So that means that nutrients which is in the middle of the football, it have to travel a long distance, reach to the surface of the football, then it can get transported outside the football, right? So that means they have to transport a long distance. But if you think about the table tennis ball, it's very small. So if if there is a nutrient in the middle of that table tennis ball and it needs to get outside the ball, it has to travel shorter distance, right? So this is that this similar kind of things are happening in case of microbes. As they are very small, both nutrients and also waste products, they can transport very efficiently and rapidly. Okay, so and eventually this helps the microbes to achieve a higher metabolic rate and generation time. Okay, that means their growth rate is very high. So thus the small size helping the microbes to generate or grow very fast. So usually higher metabolic also usually higher cellular metabolic rates and growth rates and larger population, that means high numbers of rapidly metabolizing cells, allow more efficient energy management relative to other large cells and multicellular organisms. That means microbes, they can manage the energy, okay, more efficiently than bigger organisms or larger organisms or multicellular organisms. Okay, so now if living conditions are favorable, the microbes, they can grow very rapidly. Okay, and so thus it helps them to get it helps them to prevail in the nature. Okay, so that means with a very short period of time they can colonize in a place and they can prevail there. So this was about the these were the significance of being small. Okay, so that's all for lecture 7. Thank you for your patience.